So now we will study the different characters before we study just the general properties of the retroviruses. Now the different characteristics. The most important of these characteristics, we will see that what are the salient features. There are some different features which make them different from the other viruses. The viruses present on the ground at this moment and the retroviruses, some of the retroviruses has some special characteristics. One of them we have already studied is their nomenclature, their name that they will transcribe on the backwards. That's why they are called the retro. So most challenging for the human beings so far, the retroviruses, and of course we have already seen the example of the HIV. The retrovirus structure, all the retroviruses have these things already inside them, surrounded by the viral envelope. You can see in the pictures, then they are the icosahedral in shape, contains two identical RNAs, the molecules of the RNA, which are just similar, contains the enzyme reverse transcriptase. So here you can see in the diagram, there are the glycoproteins and viral RNA, lipids envelopes, and at the same time, inside the RNA, there is the reverse transcriptase enzyme, which is playing its role at the time of the life cycle of the viruses. Virions of the retroviruses consist of the envelope particles of about 100 nanometer in diameter. These virions contains two identical and single-stranded RNAs. These RNAs are size about 7 to 10 kb in the length, which is very small. And although the virions of different retroviruses don't have the same morphology or the biology, all the virions, even if they are classified in the retroviruses, but it's not must that they will have the same morphology or they will have the same biology. They can have the different. Actually, the retroviruses again can be classified into many classes on the basis of which they will be classified later. But the most important, the components of the virions of all the retroviruses will be the same. Now the virion components, what are those components which are the same? However, their morphology or their biology will be different. These are the envelope. Of course, we have already studied the envelope of the retroviruses. All the retroviruses will have the capsid which is surrounded by an envelope. Then there is the nucleic acid, the RNAs. There are the two identical copies of RNAs are there in the retroviruses, which also contains the reverse transcriptase enzyme, which is their salient features of the retroviruses as compared to the other viruses. Then there are proteins. The different kind of proteins are always present inside these virions, which we will study them one by one. The envelope is composed of the lipids envelope, and then it is obtained from the host plasma membrane. Actually, the envelope is obtained from those lipids are not constituents of the whole virus, but they are obtained from the host plasma membrane. Then the glycoprotein encoded by the envelope gene, which is present inside. And in its function, the functions of the envelope, of course, the most important function of envelope of the virus is capsid, and it protects the capsid from the external material. Protection from the extracellular environment via the lipid bilayer. The lipids has two layers in the envelope, so it's very strong that it can protect it from extracellular environment, enabling the retroviruses to enter and exit the host cell. Again, the envelope plays an important role. It has the spikes, which it has the spikes sometimes it don't have the spikes, but it has the role for entry of the viruses inside the body through the endosomal membrane trafficking. Then the ability to directly enter the cells by fusion with their own molecules. Then with the help of the envelope, it is also the possibility that virus will directly enter inside the cell. And we already know that entry of the virus inside the host cell is very important for the life cycle to complete and to replicate to other generations. Then the nucleic acid, two identical copies of RNAs are present inside the nucleic acid, which are the dimer RNA. The cap at the 5 end and the poly-A tail at the 3 end. Of course, the caps for the initiation and the poly-A tail to terminate the codon. The terminal non-coding region for the replications are present, which are 3/8. Internal regions that encode the virion proteins. Then 5 end includes the 4 regions, which are R, U5, PBS, and L, which we will study later. The proteins consisting of GAG proteins, protease, pole protein, and the envelope proteins. We already studied that all these proteins are on the basis of the replication, and then they will give rise to different components of the virus. Now the salient features. What are the salient characteristics or salient features of the retroviruses which are making them different from the other viruses prevalent on the earth? Most importantly is their way of the replication, their retro way, the backwards way of the replication. Retroviruses are the only animal viruses that integrate to the host cell's genome. The host cell genome they will integrate, and the host cell will just take them as their own body, and they will be replicating, they will taking their food and they will using them, they will using the ATPs and energies just like the host cells and replicating inside the host, and host will take them just like their own body. The retrovirus genomes can accommodate the changes into its own configuration. The retrovirus life cycle begins in the nucleus of an infected cell. As we already studied in some of the viruses, the life cycle is completed in the cytoplasm and nucleic acid leaves undisturbed. However, for the retroviruses is concerned, all the life cycle will be completed in the nucleus of the host. Now, these salient features of the retroviruses give us a very important tool, very important research aid for delivering the genes to the target cells at the high efficiency that allows for long-term and stable expression of introduced genetic elements. These genetic elements we are already studied because we are studying in the laboratories. The scientists are doing a number of the experiments, just like we are doing the PCRs and other things. We will see in the experimental class that how we use the salient features of retroviruses and how we can apply them in the laboratories and nowadays and see the characters of the other viruses.